Chapter 23 is about population genetics, and it's a pretty important chapter in AP Bio as far as evolution is concerned. This is where you'll learn about the dreaded Hardy-Weinberg and have to learn how to calculate allele and genotype frequencies. I promise it's not that bad. This video is to introduce you to the world of population genetics and how to tackle Hardy-Weinberg problems. I'm going to make a second video for this chapter to discuss the assumptions of Hardy-Weinberg and why they don't hold up in the real world. One of the biggest misconceptions is that an individual organism can evolve, and that's totally not true. Organisms can be different from each other due to genetic variation. It's what makes everyone different. And depending on the environment that the individuals live in, a certain trait can either be selected for or selected against, which is natural selection in action. So with certain individuals being successful in reproducing, while others struggle and eventually die off, this can change the makeup of the population. Therefore, the smallest unit that can evolve is a population. The book gives an example of the ground finches of Daphne Major Island and what happened during a drought. The only seeds that were available to eat during this time were large, hard seeds. The birds with small beaks, who usually ate the soft seeds, had trouble eating and did not survive. The birds with larger beaks that could crack the seeds were selected for. So you can see in only a few years, the average beak size increased in this population over multiple generations. The population evolved, not the individual birds. You can actually break down evolution into two parts. If you're looking at the big picture, say evolution above the species level, such as cladograms and phylogenetic trees, this is what would be termed macroevolution, and we're going to learn about that in another chapter. What we're looking at in this chapter is evolution at the smallest level, like populations, and this is called microevolution. We're going to be looking at how the frequency of alleles for a trait, the number of big A's and the little a's in the population, can change over time. This can happen by a number of mechanisms shown here on the slide, but that's for the next video. So let's start by reviewing genetic variation. To have natural selection and evolution happen, you need to have variation in your population. If every organism in a population was genetically and phenotypically the same, and the environment changed so that it selected against those individuals, then you wouldn't have a population left. This variation in a population is something that Darwin observed and knew was necessary for evolution, but he didn't know how traits were actually passed on to offspring. That's where Mendel comes in, and he proposed a method of inheritance where traits are passed on through genes. We know that genetic variation between individuals is due to the fact that these individuals have different genes. Remember that alternate forms of a gene are called alleles, and that a trait is determined by two alleles. You get one from mom and one from dad. Together, the two alleles will determine the genotype and thus the phenotype. In this picture, we see a lot of variation in the coat color of horses. Coat color is actually determined by multiple genes, polygenic inheritance, just like skin color or height, which we've looked at in our genetics unit. Genetic variation provides the raw material for evolutionary change, and two main sources of genetic variation are mutation and sexual reproduction, which we've looked at in our cell division and reproduction unit. Let's quickly talk about what a population is. A population is a group of individuals of the same species who live in the same area. Remember that the biological definition of a species means that they can interbreed and produce fertile offspring. Here's an example of two populations of caribou in the Yukon, and they have a bit of overlap in their geographic area, but they tend to mate within their own population. You should also understand what a gene pool is. A gene pool is a collection of all of the alleles in a population, both dominant and recessive. So when a new generation of individuals is created, we use the alleles that are available from the gene pool. The frequency of an allele can be calculated. Let's look at a population of wildflowers that is incompletely dominant for color. The R allele codes for red, so the red flower would have RR, and the W allele codes for white, so the white flower would have WW. A pink flower is heterozygous. It would have both of the alleles of R and W. In this population of 500 flowers, 320 are red, 160 are pink, and 20 are white. How can we take this knowledge and figure out the frequency of R and W? If we have 320 red flowers, how many R alleles do we have? Since red flowers have 2 R alleles, they would have a total of 640 Rs. That's 320 times 2. And how many W alleles do we have? The answer is none. There are no W alleles in the genotype. Let's take a look at the white flowers. If we have 20 white flowers, how many R alleles do we have? Again, the correct answer is none. And how many W alleles are there? If each flower has 2 W alleles, then the total number would be 40, 20 times 2. And lastly, let's look at the 160 pink flowers. How many R and W alleles do we have? Because each flower has 1 R and 1 W, we would have 160 Rs and 160 Ws. So if we add up the total, we see that we have 800 Rs and 200 Ws. To turn this into frequencies or percentages, we take the number of R alleles and divide by the total number of alleles, and do the same with the W alleles. So we have 800 R alleles divided by 1,000 to get 0.8, and 200 W alleles divided by 1,000 to get 0.2. Notice that if you add up both alleles, they add up to 1. And we're going to come back to these numbers in a minute. So what's this Hardy-Weinberg thing? Hardy and Weinberg, these are the two guys who independently came up with the same idea. They came up with an equation to describe a population that is not evolving. This population is hypothetical and can be used to see if evolution is taking place. Any changes are due to the recombination of alleles from sexual reproduction. Remember, crossing over and independent assortment during meiosis, and random fertilization of the egg and the sperm. The premise is, if there is no evolution, then the allele frequencies will remain constant from generation to generation. This is referred to as Hardy-Weinberg equilibrium. We can also use Hardy-Weinberg to predict the genotype frequencies of future generations. All you need to do is plug in the allele frequencies into the equation. How does this work? If we go back to our flower example, the frequency of R was 0.8 and the frequency of W was 0.2. The frequencies of the two alleles are represented by the letters P and Q, and they must add up to 1. So we have this first equation, which is used to calculate allele frequencies. And while in our flower example, nothing was dominant or recessive, but usually P represents the frequency of the dominant allele and Q represents the frequency of the recessive allele. So in the case where there are only 2 alleles, then we can look at the genotype frequencies of the population as P squared plus 2PQ plus Q squared equals 1. P squared represents the part or frequency of the population that is homozygous dominant, or in our flower example, RR. Q squared represents the part or frequency of the population that is homozygous recessive, or in our flower example, WW. And 2PQ represents the frequency of the heterozygous population, RW. Please remember that in these equations, we are talking about the frequencies or percentages of the population for each genotype, not the actual number of organisms in the population. When doing a problem, make sure you look at what is actually being asked. So let's do a problem. A population has 700 individuals, 85 of genotype big A, 320 of genotype big A, and 295 of genotype little a. What are the frequencies of alleles big A and little a? Assume the population is in Hardy-Weinberg equilibrium. You're going to see that sentence a lot in all your things. When the problem is talking about a population, which equation would you use? Here you'd use the bottom one. And here's a hint. To start off these problems, the easiest thing to do is to find the frequency of the recessive portion of the population. Do you know why? If you were to find the frequency of the dominant portion of the population, you would need to take into account both p squared and 2pq, since being dominant can include homozygous dominant and heterozygous individuals. And you don't want to be solving for two variables, p and q. It's just easier to stick with finding q squared. Next, to find this, we need to find the percent of individuals who are recessive. Do you know how to do that? You take the part and divide it by the whole. So the part would be the 295 individuals who are a, and you would divide it by the total number of individuals in the population, which is 700. And that's going to give you q squared equals 0.42. Now that you have q squared, you can find q. So the square root of 0.42 equals 0.65. This is the frequency of the a allele. And now that you have q, it's very easy to find p. You go back to that first equation. It's just simple algebra. 1 minus q, which is equal to 0.35. This is the frequency of the big A allele. And if you look to see what the answer is looking for, it's just looking for the frequencies of the alleles, which you just found. So you have successfully solved this problem. My advice to you is look at what information you have. Remember, dominant traits include both homozygous dominant and heterozygous individuals. Don't confuse allele frequencies with genotype frequencies. Know which equation you need to work with. If allele frequencies are not given, then you need to look for the number or frequency of recessive traits, since that's going to give you q squared. As far as what you need, what does the question ask for? Is it looking for information on alleles or genotypes? Is it looking for frequencies or actual number of individuals? We will be practicing questions both at home and in class. The more we practice the different types of Hardy-Weinberg questions, the easier it will be for you to solve the problems. That's it for me. Here's another problem you can try on your own. We'll go over it in class. See you next time.